Thanks. I'm Kei Sato from the Institute of Medical Science, the University of Tokyo, Japan. So first of all, I'm so happy and honored to be here. But as Nakai-sensei introduced, I'm not the bioinformatician, so I'm a bit embarrassing to be here. But I'm so happy to be here and to introduce our studies mainly focusing on SARS-CoV-2 during pandemic. So let's get started. So I think most of the people have interested in our studies focusing on SARS-CoV-2 during pandemic. But before doing that, Okay, go to slide. さっきレーザーポイントも出た。レーザーポイント出しますか。はい。こっちからますよね。大丈夫です。But before starting my study on SARS-CoV-2, I just want to introduce myself because I think some people from abroad, so I just want to introduce myself and my country. So again, I'm Kei Sato. I'm 42 years old, and I'm a professor in Institute of Medical Science, the University of Tokyo. But I was born in a city called Yamagata. So do you know my city, Yamagata? Raise your hand if you know. I think most of the people are Japanese. But so I was born in the city in 1982. So this city is very small, and I think most of the people from abroad do not this city because it is very small. But when I talk to some people from abroad, actually last year I went to Singapore. Then a scientist asked me, "Where are you from in Japan?" So then I answered, "I'm from Yamagata, but you don't know. I think you don't know my city." But apparently he said, "So I know Yamagata, but I don't know why." So then we talked about the reason why he knew my city, Yamagata. Then we noticed that he is the scientist on influenza. So I think there's only a few people who are interested in biology, but the Yamagata is the name of a lineage of influenza B virus. So there are two lineages, one is Yamagata and the other is Victoria. So then he knew the name Yamagata as the name of influenza virus, not the name of city. But anyway, so I was born in the city that is associated with virus. And then I spent my time for 18 years until the end of high school. And then I went to Tokyo University in Sendai. And I spent my time for 4 years as undergrad. So, and also the important thing is Sendai is also the name of virus. So there's a Sendai virus that is included in paramyx virus. So, but anyway, so of course this is just a coincidence. I was born in a city that is associated with the virus, Yamagata. And then I spent my time for additional 4 years in Sendai that is also associated with virus. So then in these 22 years, my interest was in viruses. So then I just decided to learn about viruses in Kyoto University. So then I studied about HIV and retroviruses in Kyoto University for 13 years from a master course and PhD student and postdoc and assistant professor. So then, as Nakai-sensei introduced, I got an offer to be a PI in University of Tokyo in 2018. So then I took a job in Tokyo. Then I spent my time for 3 years as associate professor, and then I was promoted to be full professor in 2022. So then at first, I just briefly talk about my studies before pandemic. So as I said, I studied about HIV. So important thing is these human-specific pathogenic viruses such as HIV and HCV and Hep C, and these viruses can infect and replicate in humans, and that can cause diseases. So in the case of HIV, that can cause AIDS in humans. But the important thing is these human-specific pathogenic viruses cannot replicate in other animals such as mice. So meaning that it's quite difficult to investigate how these viruses can cause diseases in the body. So for that, at first I briefly talk about the HIV, human immunodeficiency virus type 1, HIV-1. So, HIV-1 is the causative agent of AIDS, and this virus can target CD4 positive T cells. So then when we got got infected with HIV, that can cause a gradual decrease of peripheral T cells, and so that can cause chronic immune activation. Then finally, that can cause immunodeficiency in humans. But again, HIV is specific for humans, so it's quite difficult to investigate how HIV causes diseases in humans and animal models. So to investigate the pathogenicity of HIV-1, so we generated a new animal model that is called humanized mice. So the recipient mice is nog mice that is established in Japan. So this mouse has congenital defect in T cells, B cells, and NK cells. So therefore, this mouse is severe immunodeficiency. So therefore, the transplanted xenogenic cells can be efficiently maintained. So then when to make humanized mice, these nog mice were eradicated and then transplanted with human CD34 positive hematopoietic stem cells. So then after 10 to 12 weeks, so this is the data. So we can detect many kinds of human leukocytes or immune cells in many tissues, such as peripheral blood, thymus, bone marrow, and spleen. Also, we can detect T cells and B cells, and also we can detect CD4 T cells and CD8 T cells. And also, the thing is, the purpose of this study is to use these mice for HIV studies. So we can detect the cells expressing CCL5 or CXL4. These are for the co-receptor for HIV infection. These can be detected in CD4 T cells. So then actually, when we inoculated with HIV-1 into humanized mice, we can see, as you can see here, we can see a high level of viremia for more than 1 year. And so meaning that we can reproduce the HIV infection in vivo using humanized mice. So using this animal model, this unique animal model, we started studying about how HIV causes diseases and how viral proteins can play a role in HIV infection in vivo. Then we published many papers using humanized mice, as you can see here. So then, as you can see here, we know that HIV has 9 genes. In the 9 genes, these 4 genes are called auxiliary proteins. Therefore, because these proteins are occasionally important for viral replication in cell cultures. So it means that some cells requires the expression of these proteins to replicate viruses, but some cells do not. So as I said, we don't know how these proteins play a role in HIV replication in vivo. So for that, we generated a gene-modified HIV-1. So I mean, we prepared we can say gene knockout HIV by reverse genetics. So we knock out the genes encoding BIF and VPU and VPL, and then we compare the phenotypes of infected humanized mice with wild-type HIV and gene knockout HIV, then try to see how these proteins can play a role in HIV replication in vivo. So I mean, so what I would say is, so I studied about HIV and retroviruses until 2020, I mean, before pandemic. And especially, I particularly focused on the roles of HIV proteins in vivo. But then, SARS-CoV-2 emerged at the end of 2020. So we started the pandemic year from 2020. So here I just show the infection cases of some Western countries such as United Kingdom and United States and Germany. So we remember that we had a huge infection cases in the beginning of the pandemic, especially spring 2020, as you can see here. But when compared to these Western countries, so we remember that, especially if you're Japanese, you remember that when compared to these Western countries, in our country, Japan, the infection cases are much, much lower than those of Western countries. That's what you can see. So this is the data from Japan. This is much, much lower than those of Western countries. So this means to control HIV, to control SARS-CoV-2 infection, I think Japan succeeded when compared to these Western countries. But when we try to think about the science on SARS-CoV-2 and COVID-19, I just try to count the number of the papers that include COVID as a keyword by Web of Science in 2020. So as you can see here, so there are many papers were published from United States and UK and Germany in Nature, Cell and Science. So as I show in the previous slide, and we know that in big cities such as New York, LA, and London, many cities in these countries are locked down to stop virus spread. So it means that it would be quite difficult for the scientists in these countries to study about SARS-CoV-2 because they cannot go to institute. But still, they have many papers. So meaning that it's quite easy to imagine that each institute and each scientist work together to combat COVID. But when I go back to the story in Japan in 2020, so as I showed in the previous slide, the impact of pandemic was not as great as in Western countries. So it means we can go to the institute. And actually, we had sort of the emergency code that's called Kinkyū Jisai Sengen in Japanese, but there's no legal restriction on behavior. So we can, so of course, I think, most of Japanese applied, so they accepted the offer from the government. So therefore, they stayed at house. They can't go outside. But we could go to the institute, even we are in Tokyo. So we can do science about SARS-CoV-2 from the beginning of pandemic. And also, we know that many Japanese funding agencies such as AMED, JSPS, and JSD, they supported studying about SARS-CoV-2 from the beginning of pandemic. So meaning that we could do many things about the studies on SARS-CoV-2 and COVID to combat the pandemic from Japan. So there's no restriction. But when I do the same thing, only a few papers are published from Japan about SARS-CoV-2 in the beginning of pandemic. So I was so surprised when I see this. So then, as I said in the previous slide, I was the expert on HIV and retroviruses. These are quite different from coronaviruses. HIV and coronaviruses are both viruses, but for the virologists, these two are quite different. So it was difficult for me, to be honest. It was difficult for me to start studying about SARS-CoV-2 because it's quite different. It should be a quite different field for me. But when I see this, I think I'm a virologist. So then I thought that so we have to do something about it as virologists. So then, fortunately, in the next year, in 2021, January, I could get a fund from AMED to study about SARS-CoV-2. But the amount of the budget was quite huge for me, to be honest. So then it was quite difficult to proceed that project by myself. So then I asked my colleagues or friends in Kyushu Island, especially Kumamoto University and University of Miyazaki, actually Dr. Aikida, Dr. Mototono, and Dr. Saito. These guys are the expert on HIV. And these guys are similar ages as me. So I knew these guys from a mass school student. So we knew each other for more than 10 years. And also I knew that they also studying about SARS-CoV-2. So, and also, I think maybe he might be here, Dr. Nakagawa, he's the informatician in Tokai University. So these four guys are my colleagues and my friends. So then after getting the budget, I asked them to join my project together. So then fortunately they said yes. So then we could start collaborative studies focusing on SARS-CoV-2. But I think maybe this might be one of the most important things for my talk today is, so, to proceed SARS-CoV-2 researches, so we need BSL-3 facility, biosafety level 3 facility. So many universities and many institutes have that facility, so we can do that. And also we could budget from these Japanese funding agencies, so we can do that. But the thing is, one of the biggest things that can inhibited the studies on SARS-CoV-2 in Japan is there are few people who can do a biological experiment in Japanese institute. So actually, so I had some students, but in the beginning of pandemic, only the people who are postdoctoral staffs can do the experiment in BSL-3 facility. So there are some students, but they cannot do that. So only staff could do that. So meaning that, so in that case, in 2020 or 2021, so I had put myself and one staff, Junpei, I think he must be here. So these two guys are the staff, the postdoc in my lab. So meaning that only, so Junpei is the infant, he do not do any experiment. So meaning that myself is the only guy who can do the experiment. So I think the similar things would happen in many Japanese institutes. Therefore, it was quite difficult to proceed the science on SARS-CoV-2 in the beginning pandemic. But again, so now I can gather my colleagues, like them, so then we can collaborate together. So meaning that if they themselves, but we can share the project. So then we can proceed one project together. So then we started this collaborative studies. So I just named that collaborative team as the Genotype to Phenotype Japan, G2 Japan Consortium. So as you may know, so we are so motivated and so productive. So we can gather many additional people from other Japanese places such as Hokkaido University. So the people in Hokkaido University, like Dr. Fukuhara, Dr. Tanaka, and Dr. Matsuno, and Dr. Sawa, these guys can do the animal experiment using hamsters. So they can evaluate the pathogenicity of SARS-CoV-2 variant using hamsters. And so I was in Kyoto, so I have some friends or collaborators, physicians. So they are like Dr. Shirakawa and Dr. Takaori. These guys are physicians, so they can gather patients here for SARS-CoV-2 studies. And also Dr. Hashiguchi is expert on structural biology, so he can resolve the structure of SARS-CoV-2 spike variant. And so Dr. Takayama he's in SAIRA, IPS Institute, and he can be expert on preparing human iPS-derived respiratory tissues from humans. So maybe this might be easy and maybe sort of take-home message of my talk today is, so SARS-CoV-2 is still continuing to evolve, but so SARS-CoV-2 evolution can be distinguished in two types that can be divided before or after Omicron. So before the emergence of Omicron, the project tree is quite diverse. So maybe you remember SARS-CoV-2 variant, alpha variant, delta variant, and Omicron variant emerged and spread. But as you can see here, these three variants are quite different. So meaning that these variants emerged completely independently. But after the emergence of Omicron, the evolutionary pattern was quite different. So the original BA1 is here, the next one BA2 is here, and BA4 and BA5 are here. So this evolutionary pattern is called small and stepwise evolution, or drift-like evolution. So the evolutionary pattern was quite different before or after the emergence of Omicron. But as you can see here, we had many kinds of SARS-CoV-2 variants. So the main purpose of our GTP Japan Consortium is to track down new emerging SARS-CoV-2 variants as soon as possible. So I think this is very small, but this is my publication list focusing on SARS-CoV-2. So we have many papers, as you can see here. But important thing is the papers shown in red here is the studies conducted by GTP Japan Consortium, not only my own lab. So as I said, we had many kinds of SARS-CoV-2 variants, such as Delta variant, Omicron BA1, BA2, BA5, and some recombinants called XBB, and new one, BA26, JN1, and something. So we, as I said, when we find, actually my staff, Junpei, he finds a new risky variant, then we can start the project. Then we work together. Then some people can do something, the other people can do the other thing. So that we can do many things at the same time. Then we can put everything into one paper on one specific variant. So we just repeated that thing for more than 3.5 years from the beginning of 2021. So as you can see here, we have many stories, but today I talk about our studies focusing on BA4 and BA5 that emerged in South Africa and spread in 2022, 2 years ago. So when we usually study about the newly emerging SARS-CoV-2 variant, we usually focus on these 5 things: transmissibility of fitness, and immune resistance, and S2 affinity, food density, and pathogenicity. So each part can be performed by each people. So as I said, we collaborate. So these 5 things can be performed by each 5 group. And then we can put into one paper. So at first, as I said, Junpei, my staff, so he found that a new variant that are called BA4 and BA5 were emerged in South Africa. So in this panel, each dot indicates the percentage of each variant. The data was obtained from a GISET public database. So based on this data, he fitted mathematical models to calculate the effective reproduction numbers, that is, the fitness of each variant. And he found that the reproduction numbers of BA4 and BA5 were 1.24 greater than that of BA2. So he found that in May 2022. So in that time, BA4 and BA5 are detected only in South Africa and some African countries. There's no cases in Japan. But based on this data, we assume that BA4 and or BA5 can be the most predominant variant in the near future. And actually, our assumption was correct. Actually, mainly BA5 was the predominant variant in the summer 2022. So as in this case, so I think, because I think, most of the people here are informaticians. So I think the biology field needs these people who can do this kind of informatic analysis. So if you have any interest, I think there are many things that can be done by informatics in the field of biology. So then we perform phylogenetic analysis, and this is the phylogenetic tree of Omicron variant. So as you can see here, the branches of BA4 and BA5 are from that of BA2. So based on this, we assume that BA4 and BA5 are descendants of BA2. And when we try to focus on the spike protein of BA4 and BA5, so there are two things. So one is the spike protein of BA4 is identical to BA5 spike. And when we compare to parent of BA2, there are 4 mutations in spike: HP69 to 70 deletion, and L45 to R, and F486B, and R496Q. So then we try to see whether BA4 and BA5 can evade antiviral immunity caused by the infection of BA1 and BA2. This breakthrough infection means the infection of SARS-CoV-2 after 2 doses or 3 doses vaccination. So we prepared the pseudoviruses with the spike protein of this SARS-CoV-2 variant. These are reportavirus. So we mixed these viruses with the Sera, who got infected with BA1, BA2. And we found that BA4 and BA5 is almost 2-fold more resistant than BA2 in both cases. And then to try to see which mutation can contribute to this immune evasion, we prepared point mutant of these 4 the mutations that were detected in BA4 and BA5 spike when compared to BA2. We inserted each mutation into BA2 spike. So then we did the same experiment. And we found that in the case of BA1 breakthrough infection Sera, this mutation, F486B, is crucial to be resistant to BA1 breakthrough infection Sera. So then we try to see the S2 binding affinity of the spike protein. So S2 is the receptor for SARS-CoV-2 infection. So this study was conducted by our colleagues in Israel. They prepared the recombinant spike receptor binding domain of BA4 and BA5 and BA2. And they mixed with S2 in vitro, in vitro. Then they found that the binding affinity of BA4/5 is similar to that of BA2. But again, so when we try to see how each mutation can affect the binding affinity of BA4 and BA5 spike to S2, so we found 2 things. One is that L45 to R mutation can increase S2 binding affinity. This KD value, the decrease of KD value, meaning the increase of binding affinity. So again, so L45 to R can increase S2 binding affinity. So on the other hand, another mutation, F486B, can decrease S2 binding affinity. So based on the data on this slide and the previous slide, we can assume that this F486B mutation can contribute to immune evasion, but therefore that can decrease S2 binding affinity. But to compensate the decreased S2 binding affinity, this additional mutation, L45 to R, was acquired to compensate the decreased S2 binding affinity. So therefore, in total, the binding affinity of BA4 and BA5 can be similar to that of BA2. So then here I just summarized our findings on the Delta variant and Omicron BA1 variant. So based on these studies, both are reported in Nature in 2022. So we found that these three parameters, the spike efficacy on x-axis and the fusion-sensitive of spike protein on y-axis, and the pathogenicity, that is size of circle, these three things are correlated to each other. So the ancestral SARS-CoV-2 is here, but the Delta variant was larger for every parameters. I mean, the spike was efficiently cleaved, and the spike was more fusogenic, and Delta is more pathogenic than original SARS-CoV-2. But in sharp contrast, in the case of Omicron BA1, it was less cleaved, and it is less fusogenic, and it is less pathogenic in hamsters. So then we try to do the same thing on BA4 and BA5. So at first we performed a cell-based fusion assay to see the fusion activity of the spike protein. So for that, we prepared two kinds of cells. One is the cells expressing the spike protein on the surface, which is shown in yellow here, and the other is the cells expressing ACE2 on the surface, which is shown in blue here. And the important thing is each cell expresses split respace. So if the two cells are fused, we can measure the respace activity. So this activity indicates the fusion activity of spike protein. So based on this assay, we measure the fusion activity of BA2 and BA5 spike. So, and then as you can see here, we found that BA4 and BA5 spike is more fusing than BA2 spike. And then we try to see which mutation can affect the increased fusion activity. Then as you can see here, L452I mutation can contribute to increase in fusion activity. So then finally, we investigate the pathogenicity of BA4 and BA5. So for that, we artificially generated BA2 and BA4 and BA5 by reverse genetics. As I said, in this time, there's no cases of BA4 and BA5 infection in Japan. So it means it was impossible to obtain BA4 and BA5 from patients in Japan. So we actually generated these viruses by reverse genetics, then we intranasally inoculated these viruses into hamsters. So then, as you can see here, in the case of BA2 infected hamsters, the body weight were very similar to uninfected hamsters. So suggesting that the BA2 is less pathogenic. But in sharp contrast, in the case of BA4 and BA5, we observed a decrease of body weight. So indicating that BA4-5 is highly pathogenic than BA2. So then finally, we performed histological staining of the lungs of infected hamsters. Then as you can see here, the percentage of the area of type 2 pneumocytes, which is indicated for the inflammation. At day 5 in BA4-5 infected hamsters was significantly greater than that of BA2 infected hamsters. So in summary, so based on our studies focusing on BA4 and BA5, now we can say that the BA4 and BA5 can be the descendant of BA2. I mean, BA4-5 are from BA2. And BA4-5 is more transmissible than BA2. And we found that BA4-5 is more resistant to BA1, BA2-induced humoral immunity. And BA4-5 is more fusing than BA2. And finally, we found that BA4-5 is more pathogenic in hamsters than BA2. So based on this study, actually this is sort of oldie because BA4 and BA5 is not existed in the world anymore. But based on this, we can learn two things. One is we can say that SARS-CoV-2 will evolve in the future. So it was quite difficult or almost impossible to stop the evolution of SARS-CoV-2 in the future. But because SARS-CoV-2 will evolve to evade antiviral immunity, and SARS-CoV-2 will evolve to increase its transmissibility. But the important thing is, some people say if the viruses can increase transmissibility, the pathogenicity will decrease. But I don't think so, because this is the case. Because, as I said, BA4-5 are the descendants of BA2. BA4-5 are from BA2 that can evade antiviral immunity, and it is more transmissible than BA2. But still, we found that BA4-5 is more pathogenic than BA2. So meaning that, so we can say that SARS-CoV-2 does not necessarily evolve to attenuate its intrinsic pathogenicity. So, but still, now from May 2023, last year, SARS-CoV-2 and COVID-19 was downgraded in Japan. I think that would be the same thing in the world. So I think maybe many people just started losing the interest on SARS-CoV-2 and COVID. But so we should note that we still have some remaining questions. So as I said, we still have the possibility to have new SARS-CoV-2 variants, such as Pi variant next to Omicron. And also we still have the problem on long COVID. And we still not sure about the duration of vaccine efficacy. So, we started the new vaccination from this month, October, in Japan, but maybe only a few people know this thing. And also, we are still not sure about the origin of SARS-CoV-2. So we still have many questions. So in these questions, so we have some project on this. So then I just want to introduce some project on some part of this. So at first, I want to talk about the findings on new SARS-CoV-2 variants. So this project was mainly led by Yu, he's the staff in my lab. So I just briefly talk about a new variant that is called KP3 and KP2.3. So this is I just summarized the mutation of newly emerging SARS-CoV-2 variant recently as a heat map. So maybe, JN1, it was the most predominant variant in the world, including Japan, until spring this year. But then that can started to diversify. They can get many mutations. Then that can named as KP2, KP3, LB1, and LP3 or something. But all of these are the subvariant of protein of JN1. So there are many mutations in the viral genome, but I think the most important thing in this talk is this mutation, the deletion at 31, sin 31 deletion that was detected in LB1 and KP2.3. So as usual, so Junpei has studied to see which variant can emerge and which variant can exhibit more transmissibility than JN1. So then in May 29th, he found that the LB1 and KP2.3 exhibit a greater fitness than JN1. So based on this, we can assume that LB1, KP2.3 are surging in some countries, in Canada, United Kingdom, and United States. So then we just start some project on this variant, KP2, KP3, LB1, and KP2.3. So further, we paired the spike protein expression plasmid, and then we prepared the pseudoviruses with this spike protein, and then we measured the infectivity. Then, as you can see here, the infectivity of KP2 and KP3 were lower than that of JN1, but we found that the infectivity of LB1 and KP2.3 was similar to JN1. So because the infectivity of LB1 KP2.3 were greater than that of KP2 and KP.3. So based on this, maybe we can assume that the increased transmissibility of LB1 KP2.3 maybe can be partly due to this increased infectivity. But then we performed neutralizing assay using human patients here who got infected with XBB 1.5 and EG5 and HK3 and JN1. So I know it's quite busy to see, but the important thing is, as I said, LB1 and KP3 can exhibit very high immune evasion activity when compared to JN1 and the other variant in all cases. So based on this, we can assume that the increased transmissibility of LB1 and KP2.3 is also due to the increased immune evasion capacity, as you can see here. So based on this, in the summer, a few months ago, we assumed that LB1 and KP3 and KB3.1.1, all of these have the mutation at 31. So we assume that these variant, the variants that have the mutation, serine 31 deletion, can be the next predominant variant in the near future. Actually, that was correct. And the most predominant variant in Japan is KP3.1.1 now. So then we started a project on KP3.1.1, and then we reported one paper in Lancet as well. And then we found a new variant, maybe a new variant that is called XCC. So that can be the most prevalent variant in the next few months. Maybe that can be the major one in the winter at the end of this year and the beginning of next year, I think. And this study was also wrapped up in Lancet yesterday. So if you have any interest, you can see. But this is reported in preprint. So then if you don't have any interest, you can find this paper from bioRxiv. So, so I just briefly talk about our studies on newly emerging SARS-CoV-2 variant. So we just plan to proceed similar studies in the future as well. But I think maybe this might be more interesting than SARS-CoV-2 is our studies on the origin of SARS-CoV-2. So this study was led by Shigeru, he's a grad student in my lab, and this study was also the collaborative studies based on GTP Japan consortium. So here I just show the phylogenetic tree of SARS-CoV-2, it's here, and SARS-CoV-2 related viruses that were detected in bats and pangolins in wild, in Asian countries, including Japan. So SARS-CoV-2 is here. So as you can see, this panel. So there are many kinds of SARS-CoV-2 related coronaviruses in Asian countries. As you can see here. And especially this one that is called Banal. This was detected in Laos in 2022. That was reported from the people in Pasteur in France. So the important thing is this strain, Banal 2236, was isolated from a bat in Laos. So this means important thing is this virus, Banal 2236, can use human S2 as a receptor. That is same as SARS-CoV-2. So therefore, this virus was isolated, and this virus was able to replicate in human cells. So this means there are many SARS-CoV-2 related live viruses in Asian countries, including Japan, that are maintained in bats and pangolins in the wild. So, and then we can make the phylogenetic tree, as you can see here. So based on this, I think it's quite easy or reasonable to assume that about the scenario leading to the emergence of SARS-CoV-2 is there are some animals like bats or pangolins, probably bats, that have the SARS-CoV-2 related coronaviruses, and then that can cause a spillover, I mean the cross-species transmission of the viruses from bats to humans by somehow. So then that can be SARS-CoV-2. So I think now we are studying about the origin of SARS-CoV-2, but I think the studies on studying about the origin of SARS-CoV-2 should be the studies that can be prepare for the next pandemic, because I think this severe event from bats or some animals to humans may happen in the near future, especially in the case of coronaviruses. So, this SARS-CoV-2 is the third cases of the outbreak of coronaviruses in the human society in 21st century. Because, SARS-CoV-1 emerged in 2002 in Hong Kong. And we still have the problem in Middle East countries such as Saudi Arabia about MERS-CoV. That's also caused by coronavirus in 2012. And SARS-CoV-2 and COVID-19 emerged in 2019. So each coronavirus outbreak occurred less than 10 years. So I think it's quite possible to assume that a new coronavirus that can be SARS-CoV-3 or MERS-CoV-2 that can emerge in the near future, and that can cause outbreak or pandemic in the near future. So therefore, to prepare for this next pandemic, I think the studies how SARS-CoV-2 can cause pandemic is very important. So then to think about the differences or similarity between SARS-CoV-2 and SARS-CoV-2 related coronaviruses, so there are some similarity and differences each other. So of course, the host is different. The host for SARS-CoV-2 is humans, but the host for SARS-CoV-2 related coronaviruses are bats or pangolins in the wild. But as I said, both of these viruses can use the same receptor for infection, ACE2. But maybe if you have any interest, so maybe the most famous difference between SARS-CoV-2 and SARS-CoV-2 related coronavirus is the presence or absence of furin-based site in spike protein. So SARS-CoV-2 has the furin-based site in spike protein, but all of SARS-CoV-2 related coronaviruses in the world do not. So therefore, I think it's quite reasonable and easy to assume that the acquisition of furin-based site may have some role in the emergence of SARS-CoV-2 and cause pandemic. So again, as I showed in the previous slide, so we found these three parameters: spike cleavage efficacy and full sensitivity and path sensitivity. So these three parameters are well correlated. So based on this, we aim to see how about the cases of bad coronaviruses, especially banana 2236. So at first, we try to see whether the spike protein of banana 2236 can be cleaved by furin protease. So this is a result on Western protein. So in the case of SARS-CoV-2, we can detect S2 band, as you can see here. So this was cleaved by furin because SARS-CoV-2 has furin-based site in spike protein. But when we delete furin-based site by reverse genetics, then if there's no furin-based site, there's no S2 band here. So in the case of the banana virus, so there's no S2 band because there's no furin-based site in banana 2236 spike. So indicating that banana 2236 spike is not cleaved by furin protease. So then we try to see the fusion activity or fusion density of spike protein. So again, in this experiment, we just only used the plasmid expressing spike protein. No live viruses in this experiment. So in the case of a SARS-CoV-2 spike, we can measure fusion activity. But when we delete furin-based site from the spike, so there's no fusion activity. So then in the case of the banana 2236, so that cannot exhibit any fusion activity. So indicating that banana 2236 spike is not fusionic. So then finally, we try to see the pathogenicity of banana 2236. So we inoculated SARS-CoV-2 and SARS-CoV-2 delta FCS, deleted furin-based site, into hamsters via internal pathway. So as you can see here, in the case of SARS-CoV-2, that can exhibit the decrease of body weight, so indicating that SARS-CoV-2 is pathogenic in hamsters. But when we delete furin-based site, the loss of body weight was very mild when compared to wild-type SARS-CoV-2. So meaning that the furin-based site is very important to exhibit pathogenicity for SARS-CoV-2. So then in the case of banana 2236, we obtained the virus from pasture. So then we inoculated that virus into hamsters. So then the data is here. The data is shown in blue here. So it is very, very similar to that of uninfected hamsters. So meaning that there's no difference on body weight between the banana 2236 infected hamsters and uninfected hamsters. So suggesting that banana 2236 is not pathogenic in hamsters. So based on this, so now we measure spike cleavage efficacy of the banana virus and fusion density and pathogenicity. So based on these data, we can plot the data on banana in this panel that should be here, because the spike was not cleaved by furin and spike was not fusionic and the virus was not pathogenic in hamsters. But then, so as I said, we wanted to know the effect of furin-based site, right? So then finally we performed additional experiment to see the effect of furin-based site. So again, so this experiment just used plasmid, spike expressed plasmid. There's no live viruses. So then in the spike expressed plasmid, we inserted furin-based site. So then we try to see whether the acquisition of furin-based site can affect efficiency. So then our assumption was correct. As you can see here, when we insert furin-based site into a bad coronavirus, that can exhibit fusion-density. So suggesting that the insertion of furin-based site into banana 2236 spike increased fusion-density. So then finally, so we can assume that scenario leading to the emergence of SARS-CoV-2 is, I think, 12036 is here, and I think I just assume that most of the bat coronaviruses should be around here because there's no furin place site. So therefore, the spike was not cleaved by furin protease, and it's not fusogenic, it's not pathogenic. But in somehow, if these viruses can acquire furin place site during spillover event from bats to humans, so then that can increase fusion activity. So therefore, that can lead to the increase of pathogenicity. So I think that can be a key impact on the emergence of SARS-CoV-2. So finally, I want to give thanks for my colleagues in GTP Japan Consortium. So as I said, we just started a consortium by 5 people, but now we have more than 10 PIs and we have more than 100 people in my consortium. So I would greatly appreciate their great effort on SARS-CoV-2 researches in the last 3.5 years. And finally, I want to thank all my lab members. Now my lab is growing up. We have more than 30 people in lab. But my lab has more than 10 people from abroad. So my lab is getting more international. So if you have any interest, as I said, I think it's quite important to collaborate. So I just noticed that the title of this symposium is the creating bioinformatics synergy across the Asia and Pacific regions. I think this is not only the region, but also the field of science is super important to collaborate bioinformatics and the other field, especially biology. That might be very important. So if you have any interest on viruses and biology, so I would be happy to have any collaboration and some works in viruses in the future. Okay, that's all my talk. So thank you for your kind attention.
Thank you, Professor Kei, for very exciting talk. So now we will accept questions. To the audience in the floor, there is, we are sorry that, but there is only one microphone there. So if you could come to this place to ask questions, or maybe you can use chat or some other online facility to ask questions, please.
Hello, I'm interested in long COVID, but it's a very complex issue because it affects many organs. And I was wondering what's your approach to study and
I think
to research
you're
it.
right. So I think long COVID is very complicated. And I think there are some animal models to reproduce long COVID in mice or hamsters. But to be honest, I'm not sure these animal models can reflect the situation in humans. So to study about long COVID, I think it would be very important to use patient information. So, but I think some people in United States, they have a large cohort to study about long COVID. But I think there's only small cohort in Japan. So I think it might be difficult to proceed long COVID study in Japan.
Okay, so currently you don't plan to go in this direction?
I do have some to try to find some biomarkers that can predict the onset of long COVID using patient samples, but just still ongoing, so we don't have any data yet.
Okay, thanks.
Thanks. Very nice talk. Actually, you told about the spike and AC2, how it's actually interaction happening in different variants. Like, so I want to know, like, have you checked about the TMPR resist2? Like, TMPR resist2, when it's binding with the spike, it's helping to open the pockets to bind with the S2, right? So, in different variants, what is the role of the TMPR? Like, how TMPR resist2 are actually behaving there?
Sorry, I can't hear. It's difficult to.
Oh, TMPR resist2. Can I repeat the question?
Please.
Okay. So, in spike and AC2 interaction, in every when you showed in every variants how it's actually binding.
Mm-hmm.
So there was another protein that's called TMPRSS2.
Yeah.
So that TMPRSS2 actually binding with the spike, and it's opening all the pockets which are actually binding with the S2. So in different variants, how TMPRSS2 is actually showing the effect on.
So I think it's a bit different. TMPRSS2 is not the receptor for SARS-CoV-2. TMPRSS2 is the protease to cleave the spike protein. So if TMPRSS2 protease cleaves spike protein, spike can be activated, then spike can fuse the cells. So what was the question about you?
Yeah, like so suppose if TMPRSS2 is not binding with the, if you see some drug design paper, so they are not targeting only spike and S2 for inhibiting the COVID-19, right? They are targeting the TMPRSS2 also, because they are telling that if we inhibit the TMPRSS2 and spike interaction, then spike pockets will not open, and then it will not connect with the S2. So then, as you told, that is that binding is happening in the spike and S2 in different variants. So, like how much effects having of the TMPRSS2 in different variants? Like,
See. I think it's quite difficult to say because, as you said, the usage of temperature is very important for SARS-CoV-2, especially exhibiting pathogenicity. But I can say SARS-CoV-2 can use another protease that is called cathepsin that are expressed in endosome. So in the case of the Delta variant, that prefer to use TMPRSS2 to infect. But in the case of most of the cases of Omicron variant, they prefer not to use TMPRSS2. They prefer to use cathepsin. So therefore, they cannot, as I show in the cases of BA1, maybe. So how can I say? The spike protein of Omicron subvariants are less fusionic. That is because they do not prefer to use TMPRSS2. So they prefer to use cathepsin. So how can I say? So based on that, I think it might be difficult to say how to use temperature is important or not. It's difficult.
I understand. Okay, thank you.
Any other questions? Let me ask a very naive question. About you mentioned about the prediction of the future evolution of coronavirus. I am wondering that if the same technique could be used to enhance the predictability of influenza virus prevalence in the future.
Thanks for a great question. So actually, my staff, I'm Junpei, and he is doing some prediction investigation on not only SARS-CoV-2 but also influenza viruses. So I think he can do that in the near future. Yes.
But so far, the prediction has not been so powerful, right?
Yeah.
That's why.
Why? I think maybe there's many reasons. Maybe one is the data is very sparse. That is one thing. And the other thing is maybe we may need some more powerful method to perform some analysis. So I think he's now doing some analysis using protein language model or something, and AI or something. So maybe this can be helpful to improve the efficacy or accuracy.
If there are any other questions, no? Okay, so then let's close this session. But before that, the organizers have prepared a token of a lecture, and there's a special gift made of an local grass, the Okinawan grass, the crop, I think.